Hello everyone, my name is Saurabh. Today video topic is about antigen and antibody. The antigens are the large molecules of proteins present on the surface of the pathogens such as bacteria, fungi, viruses and other foreign particles. When these harmful agents enter in the body, it induces an immune response in the body for the production of antibodies. For example, when a common cold virus enters the body, it causes the body to produce antibodies to prevent from getting sick. The properties of antigens are as follow. The antigen should be foreign substances to induce the immune response. Antigens may have a molecular mass 14,000 to 6 lakh Da. They are mainly proteins and polysaccharides. The more chemically complex they are, more immunogenic they will be. Antigens are species specific. The age influences the immunogenicity. Very young and very old people exhibit low immunogenicity. Let's talk about the types of antigens. On the basis of origin, there are different types of antigen on the basis of origin. First one is the exogenous antigens. Exogenous antigens are the external antigens that enter the body from outside. Example, inhalation, injection, etc. These include food allergens, pollen, aerosols, etc. and are the most common type of antigens. Next one is the endogenous antigens. Endogenous antigens are generated inside the body due to the viral or bacterial infections or cellular metabolism. Next one is autoantigens. Autoantigens are the cell protein or nucleic acid that due to some genetic or environmental alteration get attacked by their own immune system causing autoimmune disease. Next one is the tumor antigens. It is antigen substance present on the surface of tumor cells that includes an immune response in the host. For example, MHC first, MHC second, many tumors develop mechanism to evade the immune system of the body. Native antigens. Native antigens that is not yet processed by antigens presenting cell known as native antigen. On the basis of immune response, antigen can be classified as immunogen. These may be proteins or polysaccharides can generate an immune response on their own. Next one, heptane. Heptane are the non-protein foreign substances that require carrier molecule to induce immune response. Next one are antibodies. The antibodies is also known as immunoglobulins. These are large Y-shaped blood proteins produced by plasma cells. They bind to foreign particles and invade them. These particles are foreign bodies that get attacked by antibody. Antigens are foreign pathogens that invade the body and have the capability to give rise a response from an immunity system either by grouping up with a larger molecule or alone after binding with antibodies for a particular immune response. Hence, antigens stimulate the production of antibodies by the immune system. An antibody has a Y-shaped structure made up for polypeptide subunits. Each subunit has two identical light and heavy chains. The N-terminus of each heavy chain forms an antigen binding domain with a light chain. There are two antigen binding domains forming the arms of the Y shape. They are known as fragment antigen binding domains. The C-terminus of the heavy chains form the fragment crystallization domain, which helps in the interaction with the effector cell. All four polypeptide subunits are held together by disulfide and non-covalent bonds. The heavy chains of antibodies contain variable region and their constant region. Each antibody has the identical antigen binding site and differ in the antibodies. Let's talk about the types of antibodies. Antibodies or immunoglobulins are the 5 different isotypes This classification is on the basis of their H chains. Let's look the different types of immunoglobulins and their functions. First one is the IgM. IgM is the first antibody produced in the response of microbial attack by B cells. It is largest antibody and is found in the pentramic form. In circulation, it circulates in the blood and lymph and constitutes 6% of total antibody content in the serum. IgG antibody, most abundant isotype in the plasma and comprises 80% of total antibody content in the serum. It detoxifies substances that are harmful and recognizes the antibody antigen complex. It is transferred to the placenta through the fetus and protects the infants until its birth. Next one is the IgA antibody, usually found in liquids such as breast milk, serum, saliva, fluids of the intestine. IgA in a breast milk protects an infant's gastrointestinal tract from the microbial activity. It constitutes 30% of total antibody content in the serum and is divided into two subclasses, IgA1 and IgA2. Among these, IgA1 is highly found in secretions and is also called as secretory immunoglobulin. It is involved in the production of antibody by B cells. It is present as monomer and weighs around 1,80,000 Dalton. It comprises less than 1% of total antibody content in serum. Next one is the IgE antibody. IgE is present in the least amounts, around 0.02% of antibody contain the serum. These are present in the lining of respiratory and intestinal tracts and respond to allergic reaction. The function of antibody following are some of the key functions of antibody: binds to the pathogens, activates the immune system in case of bacterial pathogens, directly attacks viral pathogens, assists in pinocytosis. The antibody provides long-term protection against the pathogens because it persists for years after the presence of the antigens. It neutralizes the bacterial toxins and binds the antigen to enhance its efficacy. They also act as first-line defense for mucosal surfaces. They ingest cells by phagocytosis. Few antibodies bind the antigen present on the pathogens. This aggregates the pathogens and they remain in secretion. When secretion is expelled out, the antigen is also expelled. Thank you.